In this tutorial, we're looking at the Idealized process validation workflow. The information is based on PDA Technical Report number 42, Process Validation of Protein Manufacturing by the Parenteral Drug Association, and published in the year 2005. And you can visit and purchase that particular guide from the PDA website at www.pda.org. Let's begin. During the process development lifecycle phase of a new protein drug substance therapeutic, we will conduct a number of development studies. This will be based on process and product characterization. We will use good scientific judgment and risk assessment to make traceable decisions. We will also assess manufacturing equipment capability. Can we purchase off-the-shelf unit operations, inoculation, preparation, cell culture, fermentation, harvest, buffer preparation, media preparation, downstream processing, ultrafiltration, difiltration systems, chromatography columns, etc. Or will we have to purchase bespoke equipment? And we'll also be assessing our prior manufacturing experience to feed forward into our new development studies. During process development, we will conduct numerous risk assessments. These will be highly planned and will help us make scientific and traceable decisions regarding defining our critical product quality and process attributes, which will in turn help us to establish and define our critical and key operational parameters and their ranges. During process development, we will have process and product characterization reports. Naturally, we will have a report on stability studies of our intermediates and our final drug substance protein therapeutic. We will have statistically designed process characterization studies and statistically designed product characterization studies. So using risk assessment and statistically designed studies, we will have a clear understanding of the critical quality attributes associated with our product and the critical processing parameters associated with our process. And we will also have structure function analysis of our protein drug substance therapeutic. We will know its amino acid profile, its peptide profile, carbohydrate content, specific activity, etc. During process development, we will generate a number of process development reports that will forever be referenced throughout development, technology transfer, commercial manufacturing, and product discontinuation. These reports will summarize the rationale and documentation for the process design, parameter categorization, and process ranges. These reports will be updated and added to after each clinical production, trials 1, 2, and 3. It will document in summary table format operational and performance parameters and their associated ranges. We will get very clear raw material specifications. We get a firm understanding of our equipment requirements for the various unit operations. We will get details regarding our intermediate and drug substance stability program. And we get an overall process description. Always remember, throughout the life cycle of any pharmaceutical product, there will always be opportunity for updating the process understanding, from development through technology transfer, large-scale manufacturing, and product discontinuation. if we trend and interpret those trends, we will always have the opportunity to update our understanding of the process. We're getting very close to manufacturing our conformance batches. At this point in time, our process validation master plan for the new process and the new site and the new facility has been approved. Prior to commencing the manufacture of conformance batches, we need to ensure that all of the appropriate qualification activities are concluded. So our process validation master plan for the conformance batches will obviously link to the site and process qualification activities. We need to ensure that all our equipment is qualified. Our cell culture, our inoculation expansion, our buffer preparation, our media preparation, our cell culture fermentation, our downstream processing, our harvest, our viral inactivation, etc. We need to ensure that all our utilities, especially our clean utilities, are qualified, our water for injection, our clean steam, our purified water. We need to ensure that our facility is qualified in terms of clean room areas and our personnel are all trained. And all our raw materials are from validated and qualified vendors. We have now finally reached the stage where we can actually manufacture our conformance batches. So, in our new facility, on our new site, for example, we're going to write a process validation master plan. Like all good plans, it will give an outline of the work to be conducted and what data we wish to generate. So, we will have one or more process validation protocols that will extract data from the production record, the batch record. We proceed into manufacturing those conformance lots. It may be 3, 4, 5, 6, 7, etc. They will be manufactured in sequence consecutively. We will then write a process validation report. Assuming we have got the supporting data, we will make a declaration that the process is fit for its intended use and we can manufacture a product consistently in this new facility using this new process. Straight away, we can start manufacturing commercial batches once we have our registry manufacturing license. And then we're into a period of ongoing process monitoring. After we have completed our conformance batches, we are forevermore into a period of ongoing process monitoring. Every time we manufacture a batch, we will get new data if we properly trend that information. So we will be always updating our process understanding.